Hello, class. So today we will be learning about the synaptic transmission between the neuromuscular junction, so the neuron, and the muscle cell. So let's look at some anatomical features which involves this process. Here we have a neuron sending signals to the muscle cells. So when synaptic transmission occurs, you are basically having the nervous system controlling our muscles. So basically, this is a junction between the axon terminal, which is our lower motor neuron, and the individual muscle fiber. So think of here, this muscle over here. So there are a series of steps which allows signals to basically be carried out onto these muscle cells and allowing them to perform the neuron to control them. So the steps which follows these are very similar to that between the neuronal junction of neurons. Instead, this would be on muscles. So step 1. Just like the neurons, the action potential eventually reaches the axon terminal, and this will cause, the opening of those calcium voltage-gated channels, and we have the neurotransmitters eventually being released. So step 1. So the action potential reaches axon terminal. So similar to neuronal junction, the neurotransmitters, and usually it would be acetylcholine, and it should be acetylcholine. Actually, it's always acetylcholine, my bad. Always. So it's always. So that the muscle will always be responsive to this. So similar to the neuronal junction, the action potential will reach the axon terminal, causing the release of these neurotransmitters. So the neurotransmitters from the neuron is going to go to these specialized areas known as the motor end plate. So it's going to go over here. So let's go. Step 2. So here at step 2, we see that the chemical gates on the motor end plate will open. So chemical gates on. So the chemical gates should respond. And the way it will respond is basically causing the opening of the sodium, so on the adjacent sarcolemma, so over here. So it's not going to open here, but over here, it will allow it to open. So it will respond, right? So the specialized region will actually cause degraded potential on the motor end plate. And this is what we would refer to as the end plate potential. So these chemical gates on the motor end plate over here will basically cause a graded potential, which here on this neuromuscular junction is referred to as the end plate potential. Now, you could think of the end plate potential being like our postsynaptic potential at the postsynaptic neuron, but instead it is on the muscle. And the way that we will have these end plate potential is we would actually have the adjacent sarcolemma. Adjacent sarcolemma opening those sodium voltage. So that would actually be over here. Think of it over here. So not on the motor end plate itself, but on the very adjacent of that. So here we would have our end plate potential, which is triggered by the adjacent sarcolemma, and we always get an action potential here. And the reason for that is we want the muscle to always respond to the neurons. If not, then we can't control our muscles, and we don't want that happening. So remember, keep in mind, this is a very simplified take on the neuromuscular junction. So the end plate potential always becomes an action potential. The reason, as I mentioned, so that the nervous system, so I'm just going to put NS, always can control the muscles. Now you could think of it this way. If our nervous system does not control our muscles properly, we would have these random spasms. And that's actually what happens to a lot of people who have certain diseases, is that they can't really control their their muscle because the nervous system can't really communicate with it properly. So this is why we want an action potential to always occur here at the sarcolemma. And the way it spreads is the positive charge. So by positive charge spreading. So it's going to spread across the membrane. So remember over here, so we have our end plate potential over here, and that will always result into an action potential, therefore spreading throughout our muscle. And then, gaining control of that muscle, either causing it to be inhibited in movement or causing it to move in movement. One thing to keep in mind here is that there's actually more ACH receptors and release. What I'm trying to say here is that you can see over here at the motor end plate, there's actually more acetylcholine receptors for those incoming neurotransmitters with those types of with those acetylcholine, so that we always have these action potentials being generated. As we know, that is actually what causes excitor postsynaptic potentials in the postsynaptic neuron. It is the same idea here. We want more of these types of neurotransmitters that will cause that depolarization that will then result to an action potential. So you're going to have more acetylcholine receptors and acetylcholine release, so it can be guaranteed that there's always an action potential, and that the communication between the neurons and the muscle is constant, not random. Okay, let's look at why excitatory postsynaptic potentials and inhibitory postsynaptic potential occurs in the context of muscles. So let's say you want your bicep to, contract. So this being our agonist, you want to release a certain type of neurotransmitter. And as we mentioned prior, you want to release acetylcholine. Sorry about that. And if you want to have inhibitory signals, you may want to be the antagonist of that muscle. So let's say our tricep. So you're going to have actually inhibition, which will occur at the neuronal synapses. So there will be no action potential, no end plate potential whatsoever because you're going to have inhibition. So instead of sodium opening, you're going to have potassium opening here. So the upper motor neuron is already inhibiting to the lower motor neuron. Therefore, you don't even see any movement. So, let's say here you want your bicep to contract. This is, let's say, our excitatory postsynaptic potential. And let's say here, let's say it's glycine is your neurotransmitter. You have inhibitory postsynaptic potential. You want to go against that contraction. You're not going to have any of these. Okay, so you're not going to have any end plate potentials or action potential because you have sent this type of neurotransmitters compared to, let's say, sending this type of neurotransmitters. So what you're going to have is you will have the inhibitory postsynaptic potential occurring at the synapses. So this will occur actually at the neuronal over here. So it doesn't actually even reach onto the muscle, or even if it does reach to the muscle, you will only have, instead of the adjacent sodium channels opening, triggering that end plate potential, you instead have these, potassium opening. So you want these excitatory postsynaptic potential to occur, or inhibitory postsynaptic potential to occur in muscles, as sometimes you want to contract. So you're going to release certain types of neurotransmitters onto your skeletal muscle, or you want to be the antagonist of that and you want to go against that contraction. You're going to be inhibitory postsynaptic potential. So you're going to have certain type of neurotransmitters being released rather than the ones causing a contraction with the excitatory postsynaptic potential. So those are some things to remember of why excitatory postsynaptic potential and inhibitory postsynaptic potential occurs with muscles as well.